committee that allow me to present our work here at ISMB. So once a patient with a genetic disorder visits a clinic, many people start to working together in parallel to find out what's causing the disorder and find a solution to help the patient. It's so difficult to find out what's causing the disorder because there are too many similarities among the individuals, and also because of so many change, still there are a lot of questions that is not discovered yet, and we need to somehow find out what's causing this disorder and misfunctionality in human bodies. To find out what exactly causing these phenotypes, normally what the researchers are doing is that they're using high throughput technologies to analyze the data. By analyzing the data generated by these methods and cross-checking it with the in-house or public databases, they end up with the list of genes. Normally, this list of genes is not a short list, and there are so many genes on it. And the main question is that which one of these genes is related to our question, to the phenotype that we're looking into. To answer this question, we have to spend lots of money and time to be able to figure out which one of these genes is exactly related to that specific phenotypes. So the solution that we're offering here is a candidate gene prioritization by data fusion. As Aristotle says, the whole is greater than the sum of its parts. And it's true that if once you put all the pieces of a puzzle next to each other, you have a better picture and better overview over the question, and you can find the better answer for your questions. So in 2006, our group developed a method by using a data fusion, and they use different sources from different sources by, for example, interactions, sequencing, and functional, and they try to use this data fusion to predict which genes are associated with the phenotypes of their interest. They named the tools Endeavour. Recently, we optimized the tools and algorithm. We add more sources and more species to the tools, and also we develop a new web interface that allow users to more easily work with the system and submit their request. So to just give you a short explanation of how the system works, is system divided into 3 modules. The data entry that you define your parameters, the core module that calculate the actual prioritization, and then at the end the visualization. So to start with, you can choose among these 6 species that we define to the system. It depends on your research, what you're doing. You can choose one of these ones. We also provide the wizard summary that you can see what you submit to the system before the actual submission, and also just name the study if for later you want to track your request, if you have several requests submitted to the system. And if it's not mandatory, but if you submit your emails as well, once the result gets ready, we can send you back the link to the result. Then the next step is that to define your training gene. We're offering several ways, and we also keep updating these ways to help users to more easily define the list of their training genes. You can choose by gene name or chromosomal region, or even if you have the list of gene as a text file, you can just simply upload your text file here, and the system transfer it to our format. So sorry, as a part of our new version, as I said, we add more data resources. So far, we have added 74 data resources in total for 6 species, and for human, we have 42 data resources. which you can choose these data resources to actually calculate and do your prioritization based on these data sources. As you can see, these are fully belong to different type of data. And the last section in the data entry is the candidate genes. And again, same as the training genes, you have the same option for defining your candidate gene here. So once you submit your request, what's happened is that we try to create a model based on the training or the seed gene, we name it here, and on the data sources. And per data sources, we create a model. And then we use those models to score the candidate gene and rank them. And then finally, by simple order statistic, we integrate them and calculate the overall and global ranking for the result and return back the result to the user and visualized. To explain the result, I'm going to example of one of our famous examples. In 2010, our colleague at UZ Leuven, UZ Hospital of Leuven in Belgium, they had a question that what gene is associated with congenital heart defect. They already knew that the genes located on chromosome 6 bands 24 and 25, they are related to the different cortical phenotypes. But for that specific question, they don't have any idea which gene or genes are associated. So they end up with the 30 genes that they already know that they are associated and related to the heart defect, and then they add all the genes located in these positions. And I also rerun this analysis again to be sure with this newly added 42 data sources. Once the result get ready and return back, we use the new parallel coordinate way of representing the data. I'm not sure how many of you are familiar with the parallel coordinate, but it's a new way that you can use to visualize massive amount of data in a single format. These two, the table and the graph is fully interactive, and they are linked together, and you can play with the data, sort them and filter them, and also you can export the data as a text file for your further analysis somewhere else. Once I filter the data based on the list of candidate genes, I can see that the tab to gene is scored as the high-ranked gene associated to my question, which was in 2010. It was get cleared and approved that this was the gene that we're looking into. So far, I show you how you can use the Endeavour, but no, I'm here to convince you Endeavour result is still good and promising, and you can use it for your analysis. To do that, I use cross-validation data analysis. We download data from known gene associated with a specific disease or disorder from different sources. We divide them into several data sets. And to do our cross-validation analysis, we use one leave one out. It means that that gene that is specifically associated and have effect on a specific phenotype, we put it, separate it from the rest of genes and add it to the 99 randomly genes and try to prioritize these data set by Endeavour. What we expect was that single gene that we're leaving out must rank as the top one in our list, which was more or less was true. We calculate the area under curve for all the data set. We get the average of those values and then compare the result with the control gene set that we had. And as you can see, the result was really promising. Unfortunately, cross-validation has its own weakness to calculate. So we decided also to do one more steps for our validation. And then we go and did the time non-stamp benchmark. We use the recent CAFA strategy method, and we try to run an analysis based on two data sets, one from the data that we had in 2013, and then try to compare our results with the data from 2015. And again, you can see the result for the gene HPO association is really good. So, to conclude, we see that how we can use Endeavour. But this is not the only tool that we're offering here. Next to Endeavour, we have another tool is named Beagle. It's a text mining tool. You can use it to create the list of your genes. For the training genes as well. And also once you have the result back, you can again go to Endeavour and go to Beagle and add those genes to see whether what disease or phenotype these genes are associated with. If you want to go further and do some variant analysis, we also offer a data sharing platform. It's sharing currently more than 4,000 exome and whole genome samples. It's actually all the samples that sequenced so far in 7 centers at the Belgium. And you can generate the list of your variant associated occurred on the gene that you are interested, and then try to use our variant prioritization tools, Ecstasy, to see how much the variants are located there or associated with the phenotype that you are interested. And at the end, I would like to thank all the people who support me and help me during this project, especially my professor, Yves Moreau. And thank you very much, and I'm happy to take any questions.
Time, I thought, could you explain what Beagle does?